Can you see the presentation? Fine. So let us go next. That is next topic in that is we have seen earlier about how the replication takes place, and replication is one of the important part in the cell division. Now cell division takes place, or cell division happens in a cyclic manner. Therefore, we have got a information, or we have got a knowledge that it is also called as a cell cycle. Okay. So this cell cycle is playing important role in the cell division, and DNA replication is a part of it. Okay. Now this cell division is important. I hope you remember cell division earlier also. Basically we have two important things about cell division. That is, cell is divided to make two daughter cells, and these two daughter cells then have again grow into further daughter cell. So it is important part of the what we can say the life cycle. Without cell division, because we know that the body is formed from just two cells, okay, one from mother and one from father. Both are having a single gamete structure, and this when they come together it becomes a haploid to diploid, and this diploid cell then later makes the cell division, and these cell divisions are responsible for formation of whole human body, which has got several functions. Now when you see about anatomy and physiology in detail, so during that you might have understood that how minutely each and every organ, each and every tissue and each and every cell is having a function, and if it is not going to occur in a proper way, then how it can be disastrous, how it can be making a problem. What's system you want to think about, maybe or any like digestive system, or maybe neuronal system, any kind of system. If you see, you will find that all these systems are playing very, very important role, and because of this important role, the body is able to do the function as the body. Okay, whatever functions are there, and all these diverse functions are because of the various aspects or various things which are present in the cell. So these cells are there in eukaryotes. The cell cycle is divided into two main stages. One is interphase, and another phase is called as a mitotic phase. Okay, while in bacteria it is not like that. It is simple fission method which is taking place. As I told you, you might have seen that bacterial cell division. Okay. So during interphase, all this we have studied earlier, that is synthesis that takes place. We have seen about that how the cell this DNA is replicated. So that is all takes place during this interphase, or it while that is cell grows, it accumulates all the nutrient required for the mitosis and the DNA replication. These are three important things which are taking place in the interphase. While in mitosis, the replicated chromosome, organelles and cytoplasm, they separate into two new daughter cells, and this is how that is a mitotic phase. And, to ensure the proper replication of cellular components division, there are control mechanisms. They are called as a cell cycle checkpoints. And as I was telling, we shall be seeing it later also at the end of this topic, that these important checkpoints are playing. And if you remember, I was always telling you, many times I told you that CDK and DDKs, that is cyclin dependent kinases, they are very, very important. So they are playing very important role as a checkpoints. And after each key step of cycle, whether the cells is that the things are happening properly or not, and if not, then what has to be done. We know that there is a repair mechanism exists. So if it is there, then repairs should be carried out, and if it is not, then if everything is correct, then it will go ahead. The cells which are without nuclei, to what we call as a prokaryotes, they don't have, as I told you, they have phases like B, C, D phase, B phases, and they don't have nucleus, so we cannot see this kind of stages as we have seen here. and the B period, that is from cell division to the beginning of DNA replication. DNA replication occurs during the C phase, and the D period will be stage between end of DNA replication and the splitting of the bacterial cell into two daughter cells. And if you remember, again I repeat, it is called as a simple fusion method. Okay, fission method, sorry. So this fission takes place so that these two things are separated. Already I told you how important this cell division is, because from the single cell this or haploid cell, that is mother's and parent, mother and father, they are giving rise to a single cell that is called as a haploid or zygote, and from that it goes into a mature organism which has got hair, skin, blood cell, internal organs, and each and every cell is having some function. Okay, maybe some cells are combining together to have similar function, but in all cases there are so many diverse functions. If you want to write down various functions in very very minute way in physiological or molecular level, then a whole book will be you may need. Okay, so this is very important, that all these daughter cells they begin the interphase of a new cycle, and they have different functions, and let us see that what happens during this. So these are three phases which are taking place. First is G1 phase. We have seen this word many times when we were going for cell this DNA replication, that how in initial stages, okay, in pre stages, how this in G1 phase, how that accumulation of things takes place, and how various proteins are synthesized. The next place is synthesis or synthesis S phase, synthesis phase, and the G2 phase, which is collectively known as an interphase, and lastly is the mitosis phase. These four things are taking place, and these four are making the cell division or cell cycle is there, that is one cell, one cycle to another cell. Then the last one, the mitosis, where the cell nucleus it divides, and there is a cytokinesis, that is the material which is there inside the cell, that is kinesis is a movement. Okay, so the cytoplasm divide from two daughter cells, and all the things, organelles, then other things are get divided into two this, and then the cell membrane completes the remaining portion and the two daughter cells are formed. Activation of each phase is dependent on the proper progression and completion of the previous one. If the G1 phase is proper, then only S phase will be proper. Okay, and then it will be activated when the signals will go. If the signals, we have seen checkpoints again. So if the checkpoints confirm that everything was correct, then it will go for the next. If there is a problem in the checkpoint, if they could not rectify it, then it will result into pathological structures. Okay. The cells have temporarily or reversibly stopped dividing their cell to be in the phase that is called as a quiescence, or quietness like. Okay, so this quiescence is also called as a G0 phase. So like that, if we'll see the figure, we can see here. Okay, that is G1 is always having what we can say equilibrium, or this one transition we'll call rather. G1 can go into G0 or G1 like that. That G1 is taking a long time. Then from G1, okay, this whole yellow color, this color that shows interphase. So in cell division, see how long is the interphase. So this whole this one, the dark this color, what we say it as a saffron color. So this saffron color shows that highest time, and very smaller time is there for a mitosis. Now if we divide into this G1 S and G2, so see that maximum time the cell takes in the G1 phase. Okay, many times cell will remain in G1 phase even for lifelong. Okay, it will remain over there and it will stay over there. Now in G0 phase, we'll discuss about all these phases again. Then next thing is it goes to synthesis phase. Lastly, it goes to G2 phase and M phase. Then at mitosis, there are two daughter cells are separated. This is how the cell cycle, or it is a schematic representation. Okay. Now let us see one by another. The first one is resting one, as quiescence phase is there, gap 0. G here means gap, G0, G1, G2, that means gap 0, gap 1, gap 2. So gap 0 means this phase where the cells has left the cycle, cell is not in the cell division cycle. So these are the non-dividing cells, they will not divide at all, and they have stopped dividing. Okay. We shall see again further discussions about this. This is just a brief description. Then gap 1 is the next phase, or it is the first part of the interface. We have seen the longest period is the interface. So gap 1 is the G1 phase. In that, cell increase in the size in the gap 1. There are various checkpoints. These will control the mechanism. They will ensure that everything is ready for the DNA synthesis. So these all checkpoint, as I told you, these are the cyclin dependent kinase, and they are causing activation of various kind of cyclin A, cyclin C, etc. So these all checkpoints are there. We shall be seeing them again at the end. So when checkpoint says that everything is correct, everything is ready for DNA synthesis, then the next phase will start. That is called as a synthesis phase, S phase. We have seen it as a DNA replication. Already we have studied DNA replication. That is the part, that is the synthesis phase. Then lastly is a gap phase. During gap 2 phase or G2 phase, the gap between DNA synthesis and the mitosis is there. The cell will continue to grow. The G2 checkpoints, they will control the mechanism that will ensure that everything is ready for that cell to enter into the M phase, and then cell can divide. Okay. Then next phase or last phase, we can see it is a mitosis. The cell growth will stop at this step. No further increase in the size of the cell will be there. The cellular energy is focused on the orderly division into two daughterly cells. The checkpoints again will be there in the middle of mitosis. Particularly we call them as a metaphase checkpoint. If you remember, prophase, anaphase, metaphase, telophase, etc. So in that metaphase, checkpoints are there, and it will ensure that cell is ready to complete cell division. Now anywhere if anything goes wrong, again that will make problem in the daughter cell, and the daughter cell's anatomy and physiology both will be affected. So it may be structurally or physiologically problematic, or they are abnormal. So this is very important, that every stages body has got so many checkpoint, and despite if everything happens, then somewhere the checkpoint is not doing. After cell division, each of the daughter cell that begin the interface of new cycle, particularly the cells which are there in a growth. If you remember last year anatomy physiology, first year anatomy physiology, you might have seen that the sum of the cells, they do not divide. Okay, example neuronal cell, etc. So that means they will permanently in the G1 phase, they will not go into it. Okay, so these are how it becomes. But those the cells which are not the cell which are actually dividing, because there are some cell which are continuously dividing, or there is always division is required, or at least in some time period. You know, RBCs are they have to be at 120 days they are recycled. So there are continuous production of RBCs is going on. Similarly, the mucosal membrane, there is always wear and tear taking place. So new cells has to be formed, and therefore in that cases it is happening. So various stages of interface, they are not usually morphologically distinguished. Each phase of the cell cycle has a distinct set of specialized biochemical processes, and these processes, they are essential, or they are the part of the initiation of cell division. So first one is the G0 phase. We call it as a resting phase. Okay, and the cell cycle with this phase starts with this phase. Because the G0 has to go into G1. If any cell from G0 has to be divided, then it should go back to the G1 phase. So the cell which has to be divided, it will go to G1, and it will there, and vice versa. If it comes to G0, then can remain quiescent for long period of time. Some may be indefinitely. As I told you, I gave you the example of neurons. So a neuron may remain in G0 or G1 phase for a long period of time. Okay. Now it is very common for the cells, they are fully differentiated, because it is earlier it was believed that this G0 phase is for resting or something like that, and later it was some scientists, they have made the point that G0 phase is actually again, it is not that cells are actually resting. Cells are actually modifying themselves, and they are modifying themselves to make some change in the function, to what we call as a differentiation. Okay, so these are the cells which are already fully differentiated, and where there are less wear and tear is there, or less wear and tear is there, example neurons, then these cells will not differentiate again, and they will not go into cell cycle. Some cell which will enter G0 phase semi-permanently. Semi-permanently means they will go for G0, remain will same time for G0, and come back to G1. And once they come to G1, then they will be again enter into C cycle. Okay. So like epithelial cells, etc., they will continue to divide throughout the organism. Okay. The word post-mitotic means after mitosis, refer to both quiescent and senescent cells. Senescent means which are happening into the aging one. The cellular senescence that occurs in the response to the DNA damage. The cell which are aged, okay, and the DNA is known to be damaged. So it happens, and then for that repair and all this, the process will be there. So it is arrest in G1, that will remain for G1 till the things are happening, and if it is not there, then they will go for apoptosis. Okay, so they made the senescence make a cell's progeny non-viable. It is often a biochemical alternative to self-destruction like apoptosis. Okay, I means either there can be apoptosis, that the older cells are dying, or the older cells are made in such a way that their progeny will not be viable, and therefore no new cells will be formed. And this is one of the important aspect of the aging, that the cells which are aging, they will not able to go for the cell division, or they will not able to enter into the next phases of cell cycle. So the most important phase, the longest phase in that is the interphase. It is a series of changes that takes place in the newly formed cell and the nucleus before it becomes capable of division again. So this is called as a preparatory phase, or it is also called as intermitosis, that is between two mitosis. We call it as a intermitosis phase. And if we look into it, because mitosis is actual cell division, but if we look into it, we have seen that the interphase is a very longer phase as compared to almost 1:9 difference is there. If 9 part is interphase, then 1 part is a mitosis time wise. Okay, the interphase lasts for around, as I told you, 90% of the total time for the cell cycle. The interphase 91% here it is written. Then interphase proceed into 3 stages. We have seen G1, S and G2 phase, which is followed by cycle of mitosis and cytokinesis. Cytokinesis, again you might have studied it in first year, that is the movement of the organelles and the cytoplasm division, that is called as a cytokinesis. The cell nuclear DNA contents, they are duplicated during S phase. We call it as a replication. We have seen it. So G1, what is G1? The first phase within the interphase. We have seen it earlier also, that is briefly we have seen. That is at the end of previous M phase, that is mitosis phase. Then new daughter cell is formed, the end cell is there. Then again there is a growth is there. So there is a beginning of DNA synthesis again. This is called as a G1. So there is a gap between the next phase wise, from first mitosis to next mitosis, the gaps are there, and there is one synthesis. So gap 1 is between mitosis to synthesis, and gap 2 is between synthesis to the next mitosis. So we call them as a 2 gaps are there. It is also called as a growth phase, because all the things are growing, all the proteins are added, all the material is added which is required for the this replication, and all the proteins, we have seen so many proteins which are required for the various replications. These are sets of proteins are there. So all these proteins are synthesized, and because of that the cell is growing in the size. The biosynthetic activity of the cells are considerably slowed down, which were actually during mitosis it was slowed down. Now they will be increasing and they will be having very high rate. The duration of G1 is variable. Each different cell, even in the same body, they will have different times for G1. The cell increases its supply of protein, increases the number of organelles like mitochondria, which are important. Then ribosome grows in size, and the cell has three options. Either it has to go into cell cycle, continue into it, and enter the S phase if everything is correct. It will stop the cell cycle and enter to Z0 phase for undergoing differentiation. I hope everybody understand differentiation. What is differentiation? Anybody?
Sir, this cell will try to mature.
Put in chat box. Your voice is not clear. It's not audible. What is differentiation? Because many times this word comes, and again at many places this word will be coming when you will go for cancer and all other things. So let me understand what you know about it. No, it is not exact like that. It is not this. Let me give you example, then you can tell me. So from two haploid cell, one diploid cell, that is what it is form is zygote is formed. Okay. Now zygote was a single cell, and zygote has resulted into various organs. Okay. So whether differentiation was involved into it or it was not involved. Yes. Others can also try to answer. Involved. So whether they are proceeding toward maturity, whether it has gained maturity. Suppose the the infant has formed the body and has developed the sex organ. Okay, the sex organ developed. Will you call that they are proceed toward the maturity? So what you will call as a Anand? What you will call them as a what? How you will exactly explain differentiation? They are increasing in number. Increasing in number, is it differentiation? Deepak, you can answer, but you put it in chat box because many time the voice breaks or something happens. Yes. Taking quite much time. Okay. Actually, very simple. If you'll go for checking differentiation word, the differentiation, yes. No, Akash, it is not like that. Now the word is, the thing is, cells are specialized, means it is rather what happens is differentiation means the cell is acquiring some new function than the mother cell or than the previous one. Okay, if even if it is in a G0 phase and it is not dividing, but the cell, when it was a daughter cell, there were two daughter cells. Okay, and now these two cells are suppose differentiating, then both of these cells, they will go in G0 phase and they will acquire some new function which was not there in their mother cell. Actually, initially, while formation, both from mother cell, fine, I agreed, but this is the process, conversion from embryonic cell to specialized cell. But what exactly differentiation means, I hope you will understand. From zygote, what will happen? Zygote will have the cell division, so it will form two daughter cells. Now two daughter cell, as per our cell division, whatsoever we have seen so far, whatsoever cell cycle we have seen it, that means what? The two daughter cell will be similar to the mother cell or parent cell. So the two cells will be same. Whatsoever zygote was there, same thing it is there. Whether zygote has got any function typically known to you, either hearing, smelling, no. So what happens? Slowly it goes. So slowly this, from two zygotes, there are two cells are there. Now from one zygote, two cells are formed. Now this two cell will go into G0 phase for some time and they will acquire some new function which was not there in zygote. Okay. So this acquiring new function every time is called as a differentiation. So from that cell, when new cell will be formed, they will have this new function. Okay. So slowly one after other, when the cell division is taking place, before every cell division there is a differentiation. Okay. And if the differentiation is there, then only they will able to acquire that particular function or specialized function. Okay. So for that specialized function, it has to go for the differentiation. So we will see about that. It will continue cell cycle and enter into A space. Now let us give example. If the zygote continue cell cycle and enter A space, then every time it will produce zygote only. So that means similar zygotes will be there and it may not be an human body. Okay. Then stop cycle, enter G0 phase for undergoing differentiation. That means now we have seen that it is not clear that how many times one will go into G0. We have seen that it is not permanently, it will be after complete differentiation, like neuron, it goes to G0 phase. That is known to us. Okay. But in this case, it may be for transient period, it will go to G0 phase, modify the chromosomal structure. Modify the chromosomal structure means just some of the DNA which were not earlier expressed, this DNA will be expressed. They will cause production of some of the enzymes, protein which were different than earlier cell. This protein will make some changes, and because of that, the next progeny of that, next cells from that, when the cell division is there, they will acquire that particular thing. So like that, some tissues are made. Okay. From this zygote, all different tissues are made, epithelial tissues, muscular tissues, connective tissues. So one after another tissues are made, osseous tissues. So these tissues are made. This is also differentiation. Then from tissues, then some of the things are there. So each and everything is happening, and this is what differentiation is there. And the next thing which can happen to that is, during G1, the cell will become arrested in G1 phase. Either it may enter G0 phase, or it can re-enter into it. And just like, hibernation, or in microbiology you might have studied that they go into what we can say, formation of example, Mycobacterium tuberculosis, that or such kind of bacteria. They will hibernate themselves and they will not allow any type of growth or something. So it will remain there in G1 phase. So it may go, if it is no differentiation only, it will remain as such. Okay. So the deciding point is called as a checkpoint or restriction point. I told you this is important one because so many times this word is coming. Every time there will be checkpoints, and there are a variety of proteins and enzymes which are playing role as a checkpoints. And these checkpoints or restriction points, they are also called as a start, and they are regulated by G1 GS cyclin. If you remember cyclin dependent kinases or CDKs. So these cyclins, they will check, they will make the check, everything is fine, they will allow accordingly the kinases will be there, this kinases will cause this, and then there will be transition from G1 to S phase and passes through G1 checkpoint. There, if G1 checkpoint is says it is okay, it is permeable, then it will go to the next phase, that is known as a S phase. S phase, you have seen it, it is a DNA synthesis. That is, from one DNA it makes two DNA to what you call as a replication. So when it is complete, the chromosome they have been replicated. The chromosome consists two sister chromatids, and during this phase the amount of DNA in the cell has doubled, isn't it? Because the parent one and the daughter one. So there are two sets are available of chromosome to what we call it a system. Though the ploidy and the number of chromosomes are unchanged, the number of chromosomes will remain same. If it is 23 pairs, the pairs will remain same. Okay. And the rate of RNA transcription and protein synthesis is very low during this phase. Why? Because DNA is engaged in replication. So simultaneously DNA cannot be processed for two different things. One is processing for DNA replication and other is for DNA transcription. So it is not happening. So it will be lesser, and only replication will take place. Only exception to this is the histone production, because it will be required for the winding up of the DNA, and therefore it will be there, which will be occurring during S phase. Last phase is a G2 phase. It is also a growth phase. The first one also growth, second was also growth, and G2 actually during S phase also there is a growth because chromosome number has increased. Okay, so therefore because of the more chromosomes or chromatids are there, it is there. So it occurs after DNA replication. It is a period of protein synthesis and rapid cell. That means again here now DNA to RNA or transcription and everything is increased. So period of protein synthesis is there, rapid cell growth is there to prepare cell for mitosis. During this phase, microtubules, they are begun to reorganize from a spindle, okay, that is called as a prophase. So they will be there. Now before proceeding to the mitotic phase, the cell must be checked at the G2 checkpoint for any DNA damage within the chromosomes. G2 checkpoint is mainly regulated by the typical protein that is called as a tumor protein, that is also called as a P53. P53 is having so many names actually. It is also called as a modulator of apoptosis. Then it is also one of the component in the pro-apoptotic protein, that is BCL2 binding component. Now these are various genes and various components which are playing important role in the cell cycle, and they are playing very, very important role in cancer. Okay, so if DNA is damaged, P53 will either repair the DNA or trigger the apoptosis. Very simple. And if this sensing goes wrong, it may trigger the apoptosis of good cells. Okay, or it may not do the DNA repairing properly and same cell continue to grow. If P53 is a dysfunctional or mutated, cells will damage, DNA may continue through the cell cycle leading to the development of the cancer. So when you study cancer, at that time we will understand how the P53 and these all BCL2 and all proteins and genes are playing important role. We will have this, that is called as oncogenes, proto-oncogenes, etc. Okay, so these are the important regulator or checkpoint is there. And last phase is there, that is called as a mitotic phase. We know about it, I'll not go into more discussion about it. There are various phases: prophase, prometaphase, metaphase, anaphase, telophase. These all phases you have seen. Okay, this is also called as a karyokinesis. Okay, and it is very short one. We have seen 91 percent time is interphase, and only 9% or around 10% time is mitosis. And during that also these all phases are taking place. These are the schematic representation one after another, because they are so fast that we cannot exactly we can say this is this phase, this is this phase, this is this phase. But these are schematically explained that in prophase what happens is this two centromeres, they go away from each other. From that mitotic this spindles are formed, that is that microtubules are extended from that, and this result into mitotic spindle formation. And then chromosomes are separated with these spindles, they are attached here. There are many things, if you remember, there are these microtubules, and they are acting as a channels also with that various organelles, and they are floating just like a rope train. Okay, so this rope train like situation is there. This rope train is carrying all organelles or genetic material, and it segregates, and lastly in it will be then making a cell division. So it is a process by which it separates chromosome in its cell nucleus into two identical sets of two nuclei. During this process of mitosis, the pair of chromosomes, they are condensed, they are attached to microtubule, and they will pull the sister chromatid to opposite side of cell. As I told you, just like a rope train, or just like a, wired cable is there which is causing pulling work. It occurs exclusively in eukaryotic cell, but various cells and various organisms have a different way. Okay, animal cells can go and open mitosis where the nucleus open up, so no nuclear envelope will be there at the time of cell division. But the fungi, there will be closed mitosis. That means nuclei will be intact, nuclei will not change, even nuclei will have its own division. So this is how it happens. Okay, in bacteria we have already seen there is nothing like this. It is simply fission is taking place. That is, only two things are there. We have seen this B, C, D phases of it. Then cytokine phase is there, that is separation of cell component. As I told you earlier, we will not go into more details. That these microtubules, they provides as a channels, and through that the materials, they are polarized to one pole to other pole, like that. Once it is there, then the cytoplasm will have that separation, and then mother to two daughter cells will be taking place, and these are genetically identical to each other, and parentical cell. So this is very, very important, that cell division always result into similar mother cell and daughter cell, while when it goes to differentiation, then after differentiation its daughter cell will be similar to the mother cell or parent cell. Okay, the cytokinesis always occurs along with the mitosis, and it is often interchangeably used, but they are different actually. Okay, mitosis is actually the cell division, cytokinesis occurs separately, which are forming single cell with a multiple nuclei in the process, that is called as endoreplication, and it occurs most notably in the fungi and this other such other organization. Errors in mitosis can result into cell death through apoptosis, or it can cause mutations, and that may lead to cancer. So why you are studying this is, the reason is this, okay, that these mutations that may lead to cancer or any kind of mutation, whenever it will be there, just keep in mind that whenever these mutations are there, that means that there is a change in the genetic structure which is something different than the normal one, if that particular thing, that particular species is fortunate, then may not impact much, the impact may be smaller one. But if not fortunate, then anything can happen. The malformation of body, malformation or malfunctioning rather we can say. So function wise or structure wise, any changes it can lead to. So thus the mutation can make the problem. Now we have seen various times checkpoint. What are these checkpoints? So regulation of this this cell cycle, that is involve a process very important for the survival cell. So it detects any genetic problem and it repairs it. Okay, and if this works properly, then it is one of the best thing which is happening, and normally it happens. If it goes wrong, then we have seen that it leads to formation of faulty pieces, and these new cell which are faulty, that can either having problem in function structure, or they can die early. Anything can happen to this. Okay, the molecular event that control cell cycle are ordered and directional, and each process occur in a very sequential fashion, and it is impossible to reverse the cycle. Once, example, if from G1 it enters to S phase, then it cannot go into G1 phase. अरे यार वो गलत हो गया था, अब मैं इसको correct कर लेता हूँ. No, the time was given, there was checkpoint, it was there. If checkpoint has made it okay, then it will go into S. Once it makes S, then it will go to the next. Okay, so this is very important, and these things are very important for a normal cell division and formation of normal cell. There are two things are there which are playing important role. As I told you, these are cyclins and cyclin dependent kinases. So these cyclins are proteins, and on that there are kinases. We know kinases. Kinases are actually activators. These are the enzymes which activate by one or other ways. Okay, we have seen protein kinases and many such kinases. So by any mechanism, these are causing activation of this, and these enzymatic activities of these cyclins, they will determine the cell's progress through the cell cycle. And two scientists, Paul Nurse and Tim Hunt, they got Nobel in 2001 for this, and this is very, very important. Most of the work nowadays, particularly related to cancer and some many other activities, is actually being concentrated on this area also, that can we do this something, because normally body has not programmed for any kind of cancer or any kind of damages, and body has made a very nice system of checking the problem and making the repairs. And this cyclin CDKs, which are the checkpoints, if they are doing it properly, then we should not have any problem. But if it is not happening, then it will be making a problem. So there are various activities which are there, and these are forming the regulatory subunits. Cyclins are forming the regulatory subunits, and these cyclic dependent kinases, they are the catalytic subunits of the activated heterodimer of these cyclins. The cyclin have no catalytic activity, but once they are activated, the cyclin dependent kinases are also inactive in absence of partner cyclin. But when both come together, if you remember, just I'll give you the analogy. Example, we have seen that acid itself is not doing any kind of digestion work, and the pepsinogen is not doing anything. But when they are coming together, HCl and pepsinogen, they will causes conversion of pepsinogen into pepsin, and then it is produced, it is responsible. Or similarly in clotting factors you might have seen. So similar thing is there. As such, CDK, that is kinases, they do not do anything, and cyclins do not do as such anything. But when they come together and when they are activated, then they will make the activated part. This CDKs will do the phosphorylation of the cyclins, and because of that it will cause inactivate or either activate or inactivate the target proteins, and they will cause the orchestrate the coordinated. Orchestrate is very nicely used word. You know orchestra. What happens in orchestra? The music plays as per the rhythm. Okay, if any music goes wrong, then you will not like it. Okay, if the music have any kind of change, so this orchestrate the coordinated entry, that means very properly managed one after another, no single mismatch into that. If in orchestra if there is something happens, आवाज़ कहीं आ रही है, एक ऑर्गन कहीं और बज रहा है, दूसरा जो म्यूजिकल इंस्ट्रूमेंट है और कहीं बज रहा है, then you will not enjoy, you will find it rather distracting instead of enjoying. But if everything is happen in a very clear way, one after another, and then you will enjoy. So similarly, it is a coordinated things are there. The different cyclin CDK combinations are there. They will determine the downstream proteins they are targeted. CDKs are constitutively expressed in the cell, whereas means CDKs are present in the cell. Constitutively means we have got two things. I think last year we have seen about it, prostaglandins, isn't it? Constitutively and the inducible, okay, or nitric oxide, constitutive and inducible. So they are constitutively expressed in the cells, whereas cyclins are synthesized at specific stages of the cell cycle in response to the various molecular signals. So what happens into that? There are several things they are happening upon receiving a pro-mitotic extracellular signal. G1 cyclin CDK complexes, they become active, they will prepare the cell for S phase, promoting the expression of transcription factors that will in turn promote the expression of S cyclins, that is the cyclin which are playing role in the synthesis phase, and of the enzyme which are required for the DNA replications. These enzymes will be prepared during G1 phase. The G1 cyclin CDK complex will also promote the degradation of molecules that function as S phase inhibitors. So any molecules which are playing as a inhibitor of replication, these will be degraded because the enzymes will be produced for the degradation of that by targeting them for the ubiquitin. So ubiquitin is one of the protein. When it is attached to any molecule, then that gives signal of its damage. So this and that process is known as a ubiquitination. You might have studied it in biochemistry. If not, you just refer it. Or if you are using Wikipedia, as I told you, you click on this blue line and you will find that what it is. So it is a small protein, and that will there, if it is attached to any particular component, then that gives signal of its damage. Once the protein has been ubiquitinated, its target proteolytic degradation by proteasome is there. So this is sort of sign, है ना, जैसे लड़ाई में कहाँ बम मारना है, वहाँ अगर कोई जाके मार्क करके आता है कि दिस इज द प्लेस विच इज टू बी टारगेटेड. So this is ubiquitin is acting like that. It is acting as a marker, and then we can target it specifically for that. Okay. The result from the recent studies shows that the single cell level argue that role of G1 cyclin CDK activities. It shows that there may be more than one type of mechanisms are there. Then active S cyclin CDK complex, they will phosphorylate proteins. They make pre-replication complex. We have seen yesterday during replication. They are assembled during G1 phase, and then there will be replication origins are there. We have all seen these all activities yesterday, and thus one by one the activities will be there. It will ensure that every portion of the cell's genome will be replicated once and only once. Okay, no duplicate pair or no some of the molecule are formed. So the reason for prevention of gaps in replication is there because of the daughter cell. They are missing all or part of the crystal gene will die. If in that, if any portion is not properly there, or if it is not proper one, then that cell will not able to do the function, and maybe if it is important for its own life of the cell, then the cell will die. So for that, the checkpoints will be ensure that everything is there. And for the reason related to gene copy number effects, possession of extra copies of certain gene is also deleterious for the daughter cell. Okay, because it will give the confusion signal. Why additional numbers? So it should be matched with the mother cell. It should be exactly like that, and therefore these checkpoints will ensure that there are no less or no more genes are there, and this will ensure that the daughter cells are healthy enough. So next to synthesis phase, then again there will be checkpoints, that is mitotic cyclin CDK complexes. They are synthesized but they are inactivated during S and G2 phase. They are promoting the initiation of mitosis by stimulating the downstream proteins which are involved in the chromosome condensation, and they will form the mitotic spindle assembly. I hope you remember mitotic spindle. If not, go through it again, because we shall see it in cancer. A critical complex activated during this process is called as a ubiquitin ligase. These ligases, they are known as anaphase promoting complexes, and these are promoting degradation of structural proteins. They are associated with the chromosome kinetochore. Kinetochore, again, these are the molecules which are responsible for kinesis, that is movement across the microtubules, and these will also target the mitotic cyclins for degradation, ensuring that telophase and cytokinesis are happening in normal way. So there are many actions these are doing. What are the actions of cyclin and CDK complexes? So this is cyclin D is the first cyclin that enters cell cycle in response to the extracellular signal, that is growth factors are there. This we shall study in the next this one various pathways we shall be seeing. Okay, so in pathways we shall see example, if you remember, just for your revision, I'll tell you example. You have studied growth factors. Where you have studied? Where you have studied in the blood cell formation. There are various growth factors are there which are causing the synthesis or the formation of various blood cells, isn't it? So these are for this these growth factors are there. Are many growth factors are there in body. Okay, and these growth factors are responsible for particularly cell division in that particular area. Okay, so this cell division will be taking place in that particular area. So these growth factors will be again released. If you remember, we have seen immunology recently. So we have seen that interleukins and all, they are playing important role, and they are causing increase in the formation of some of the cell. Example, we have seen that T cell, B cells are there. So that means they are acting as a growth factor for that, and because of these signals, that is growth factor signals, because of whatsoever signals are there, the cell division is initiated. Okay, so this cyclin D is the first cyclin produced in the cells that will enter the cell cycle. This will level stay low in the resting cells and which are not proliferating, but as soon as cell decides or cell plans, why cell will plan, as I told you, either external or internal, or internally the timers has been set. Okay, just like in we have seen in case of females in menstrual cycle. So the timers are set that after this will happen, after this will happen. So new oocytes will be formed, these oocytes will be growing, that is growth is also there. Then after some time they will be destroyed, and again after that from the primordial follicle new cells will be forming. So this is no other outer cell is no outer this one this there. Here growth factor wise what is happening is the hormones is coming in a cyclic manner. So these are the external factors which will be making it, or some time bound changes will be there, and accordingly it will be there. Then CDK4, 6, CDK2 are also inactive, because I will not go into each and every protein into it. So these all will one by one they will cause. There are various important proteins are there. They are known as a retinoblastoma proteins or RB proteins. They are also playing important role in cancer. And this unphosphorylated RB tumor suppression functions, they are including in cell cycle exit and maintenance of G0 arrest. So these are related to aging of the person, that if it is these CDK complexes, if they are monophosphorylizing this retinoblastoma or RB protein, then these proteins are doing job. And if these are and if these are there, unphosphorylated are there, then it will cause cell cycle exit and maintenance G0 or arrest into that, which is happening into senescence, that is in the aging we have seen. So there are many researches going on. Will not go into details about it, how they are playing. There are many details are there. That much details are not even required for that. We will discuss this. You just, if you wish, you can go through that. How these are there. There are various genes are for each type of this: cyclin E, cyclin A, cyclin B. Okay. Then DNA polymerases, thymidine kinases, so many enzymes are there, and they are responsible one after another activities. Okay. Now there are certain inhibitors. If you remember, we have seen the word that these are acting as a checkpoints, but there are various inhibitors also, and these inhibitors are responsible. They will not allow the cell cycle to happen. They will try to arrest it at one or other, at least G1 phase. Okay, because once G1 phase crosses, then it has to go for complete cell division. Then it has to reach to the mitosis completely. But there are many proteins are there, or many factors are there which are causing there, and they are called as a tumor suppressors. They are either genes or proteins. Okay. There are variety of this. Example of these are CIP, KIP family. They are interacting with the CDK, so we call them CDK interacting, or kinase inhibitory. KIP means kinase inhibitory, or CDK interacting proteins are there. They will inhibit these CDKs or kinases, that is CDK kinases, and because of that they will not allow the progression of cell cycle. And these are prevention of tumor formation. Tumor means what? Too much cell division. So they are also called as a tumor suppressors. Okay. So these are inhibitor are very important. There are variety of genes, and because of the genes, whatsoever proteins are formed, there are many such genes, and that gene form proteins are there. Example of these are called as a P21, P27, P57. This is depending on the amino acid which were there, and they are named accordingly, that the protein is having amino acid 21 amino acid protein with 27 amino acid protein with 57 amino acids are there. These are typical proteins are there, and they will stop the cell cycle in G1 phase by binding to the inactivation of cyclin CDK complexes, and thus they will be playing important role. P27 is activated by transforming growth factor beta, that is TGF beta is a growth inhibitor. Then there is one more family is there, that is INK4A or ARF family, that includes PIP16 which is bind to CDK and arrest the cell cycle in G1 phase. I tell you, rarely it goes beyond G1 phase, because in very few conditions it can be there, but there are some which can inhibit into synthesis phase also. Rarely they will be useful to arrest cell cycle and useful in the anti-neoplastic and anti-cancer agents which can be doing. I told you why we are using, why we are studying this. So these are there. There are many such agents are there. So there are many inhibitor of CDK25 and other things are there. Many human cancer, they possesses the hyperactivated CDK4/6 activities. We have seen that this CDK is important. CDKs, then MCMs. Last time we have seen. So these all proteins are there which are playing important role in the DNA replications, so that they will be arranging various enzymes which are playing important role into that. That is DNA dependent transperases and ligases, and all these enzymes are there. Topoisomerases, all these enzymes are important. So these are there, and this CDK4 and 6 proteins, they are having activities with cyclin D type of proteins, and inhibition of this will result in the prevention in the malignant tumor from proliferation, because Until and unless these DNAs are replicated, how the next stage will go into. So scientists have tried to invent a synthetic CDK4/6 inhibitor in that, which is having the anti-tumor effectiveness and which will be comparatively safer. When we shall go for cancer, we shall see that why the anti-cancer treatment is very what we can say, having too much adverse effects. Okay, so these are known as palbociclib, then ribociclib, then abemaciclib. These are recently approved FDA approval for clinical use to treat advanced stage of metastatic hormone receptor, that is HR positive or HER negative breast cancers. And this is orally active. These are very new drugs. These are recent drugs which are having comparatively lesser adverse effect than the previous one, and they are comparatively very specific. Okay, so these are how, and this is how the importance of studying this science. The remaining thing we shall see, that is checkpoints and all, briefly we shall go about it next time, and various other agents we shall see in next time. I hope you are understanding this, and see that you are reading to it. That will help you. Okay, make the notes of it, make the points, and make one framework of it, because it is, as I told you, it's a very science. Please keep the notes ready for you, that whatsoever has been taught, and try to make circulation of it whenever possible. So here we stop.